All right, so welcome. Thanks for joining us. My name is Elise Hoover. I'm the hospitality host for this session. This is the research breakout session. I'm very pleased to introduce the speakers for this session. We do have 3 presentations, so I'll introduce each speaker right before their talk. Our first presenter is Alexandra Hayward. She's a collegiate translational medicine student at the University of Chicago, pursuing a bachelor's in molecular engineering and a minor in data science. As a fellow, she researches racial and ethnic disparities and the impact of socioeconomic status and geography in PKD care and outcomes in the US within the section of nephrology at University of Chicago Medicine. Alexandra is also a research assistant for the Hospitalist Project, where she interviews patients about their health to improve the quality of care at UChicago hospitals. Alexandra, take it away.
Thank you so much, Elise. I'll share my screen. Okay, wonderful. Everyone can see?
Yes, we can.
Okay, wonderful. So hello, everyone. Welcome again to the research session. I am honored and grateful to be here with you all today. Thank you so much to the PKD Foundation for giving me the opportunity to share my research in racial and ethnic disparities in PKD at this year's PKDCON. So like Elise mentioned, my name is Alexandra Hayward, and I'm a collegiate translational medicine program fellow and rising senior at the University of Chicago. I'm really excited to share with you all what we found, as we hope that our findings will provide insight into reducing disparities in PKD nationwide. With over 3 million cases across the world, PKD is the most common inherited kidney disease. And in the US, incidence, prevalence, survival rates, and age of kidney failure in PKD are all increasing. As we can see here, which type of mutation a patient has significantly influences at what age they reach end-stage renal disease, also known as ESRD or kidney failure. As the 4th leading cause of kidney failure, PKD represents 6% of all patients in the US receiving renal replacement therapy. Research has shown that patients whose first kidney failure treatment is receiving a preemptive transplant have much better outcomes than those who begin with dialysis. Additionally, patients who start on dialysis but eventually receive a kidney transplant also have better outcomes than those who remain on dialysis. PKD is shown to not favor any race or ethnicity, and the distribution of genetic mutation in PKD is also consistent across race and ethnicity. We know that race is a social construct and that PKD is a genetic biological disorder. So although research has shown that racial and ethnic disparities in kidney disease exist, the impact of these disparities in PKD is unclear. Because patients often know PKD is present in their family, kidney failure is highly anticipated. However, we don't know whether disparities still exist nonetheless, so we were interested in what other factors may still lead to racial and ethnic disparities in PKD outcomes and care. Therefore, we aim to investigate the role that race, ethnicity, socioeconomic status, and geography play in age of kidney failure and access to kidney transplantation in PKD. The patients in our study population consisted of US PKD patients greater than 30 years old and who had developed kidney failure between 2000 to 2018. We divided our population into 3 categories: non-Hispanic African American patients, Hispanic white patients, and non-Hispanic white patients. We built linear regression, logistic regression, and Cox models to investigate the age of kidney failure of African American and Hispanic patients compared to that of white patients. Also, access to preemptive transplant by evaluating African American and Hispanic patients' odds of receiving a preemptive transplant compared to that of white patients. And finally, the differences between the rates of transplantation for African American and Hispanic patients versus white patients. All our models were adjusted for sex, insurance status, employment status, albumin, and hemoglobin. So in the US PKD population, African American and Hispanic patients reach kidney failure earlier than white patients and well earlier than the average age in the US of 57 years. Larger majorities of African American and Hispanic patients also start on dialysis instead of receiving a preemptive transplant compared to white patients, with almost 1 in every 5 white patients receiving a preemptive transplant, while only 1 in every 20 African American patient does. We also see that Hispanic patients receive slightly more preemptive transplants than African American patients. Overall, African American patients receive the least transplants, as the variable any kidney transplant here includes both preemptive transplants and transplants after starting dialysis. More white patients also have private insurance than African American and Hispanic patients, and we can see that employment levels from 6 months prior to kidney failure to the time of kidney failure drop at higher rates for both African American and Hispanic patients than white patients. So we found that African-American patients reach kidney failure 2.5 years earlier than white patients, and that Hispanic patients reach kidney failure 4.8 years earlier. We also found that African-American patients are 69% less likely and that Hispanic patients are 54% less likely than white patients to receive a preemptive transplant, which again, research has shown leads to better outcomes than starting on dialysis. We found that the rates of receiving a transplant after starting dialysis are also 46% lower in African-American patients and 28% lower in Hispanic patients than white patients. This figure here shows the rates of transplantation after starting dialysis by race and ethnicity, and the y-axis shows the percentage of patients who are transplanted over time. So we see that African-American and Hispanic patients receive transplants at much slower rates than white patients, with African-American patients receiving the lowest rate of transplantation. We also see that the rate of transplantation for Hispanic patients is in between that of white and African-American patients. We also investigated whether geography plays a role in access to transplantation. So in the US, data on dialysis and transplant care is reported through 18 ESRD networks defined by region, which you can see here. And so we provided an initial racial and ethnic breakdown of each network to characterize the PKD population that each is serving. We generated a color map that visualizes the odds of an African-American patient receiving a preemptive transplant compared to a white patient in each network. So here we see that lack of access to preemptive transplants for African-American patients persists across the US. An African-American patient is generally only about 15 to 35% likely to receive a preemptive transplant in any part of the US compared to a white patient, with the least likelihood in New York and New Jersey. Our results for Hispanic patients were difficult to interpret because several networks lacked sufficient Hispanic patients for significant analysis. These networks are indicated here by striped colors with their results grayed out. In fact, within the regions representing the Mississippi and Southeastern Kidney Council networks, number 6 and number 8 shown here, no Hispanic patients at all had ever actually received a preemptive transplant during the timeframe that we studied. However, in every network with significant patients, we found that a Hispanic patient is about 30 to 40% likely to receive a preemptive transplant compared to a white patient. These networks are indicated here by solid colors. Off, Hispanic patients are also disadvantaged in some regions. However, larger confidence intervals whose brackets cross the line of equivalence can be attributed to fewer data being available for Hispanic patients within our population. So compared to white patients, rates of kidney transplantation after dialysis are lowest for Hispanic patients around Washington, New Jersey, and Colorado. Thus, among all the demographic and socioeconomic factors we explored, we found that race and ethnicity, geography, private insurance, and low albumin and hemoglobin levels most significantly affect age of kidney failure, access to preemptive transplant, and rates of transplantation between African-American, Hispanic, and white PKD patients in the US. And in conclusion, our research has shown that in PKD in the US, African-American and Hispanic patients reach kidney failure earlier, have less access to preemptive transplantation, and have lower rates of transplantation than white patients with some geographic variation. Based on our conclusions, we propose initiatives, policies, and further research of the racial and ethnic inequalities in PKD that we found, including legislature allowing for the start of Medicare coverage once a PKD patient is eligible for kidney transplantation evaluation, rather than only once a PKD patient reaches kidney failure, improved education about living donation for PKD patients and their families, improved care and management of African-American and Hispanic PKD patients with secondary causes and comorbidities, and increased awareness of the genetic nature of PKD and its potential impact on family members. And finally, of course, further work to identify additional factors and biases that help us better understand the impact of race and ethnicity on PKD outcomes and care. So that concludes my presentation of what we found thus far about the racial and ethnic disparities that exist in PKD. I'd like to thank my mentors, Dr. Arlene Chapman, Dr. Rita McGill, and Dr. Milda Saunders for making this research possible and supporting me throughout every step of the way. And thank you all so much again for your time and attention, and to the PKD Foundation for the opportunity to share my work, which we hope will inform policies aimed at reducing these disparities nationwide. I'd be happy to answer any questions later in the Q&A of the research session. Thank you all so much again.
Thank you, Alexandra. That was fantastic. All right, next we have Dr. Harrison Kim. He's an associate professor in the Department of Radiology for the Division of Advanced Medical Imaging Research at the University of Alabama at Birmingham. His research vision is to globally standardize quantitative imaging of various diseases, including PKD, which will facilitate automatic prognosis and therapy monitoring for patients. His research mission is to develop hardware and software tools along with this endeavor and validate those tools in multi-institutional clinical trials. Dr. Kim?
Yes, ma'am. Okay, did you see it well?
Yes.
So thank you very much for your introduction. Today I would like to talk about novel prognostic imaging biomarker that we developed based on individual cystic growth rates in ADPKD. Let's start with some background. Height-adjusted TKV, HTTKV is most widely used as prognostic imaging biomarker, as all we know, for renal function outcomes like GFR change in ADPKD. However, it was correlated only moderately with future GFR change. For example, HTTKV explained only 35% of GFR change in the CRISP cohort and only 22% in the HOLD cohort. Besides, accurate assessments of the therapeutic response based on total kidney volume changing may require many years follow-up. Therefore, we were motivated to develop a more sensitive alternate for the disease monitoring. So we hypothesized that perhaps the index is reflecting the growth characteristics of individual cysts might correspond to the underlying disease, ADPKD activity, and more accurately than total kidney volume-based analysis. Let's take a look at our representative images. This is the kidney image of a patient in CRISP cohort, and the baseline imaging and yearly follow-up of 3 years. Some of the cysts grow faster than the others. Sometimes new cysts formated, sometimes cysts disappear over time. So we observe the differential growth rate of the individual cyst. So we may be able to create a histogram of individual cyst growth rate, for example, from baselines to 1 year. And then if there are more faster growing cysts in the kidney, then in the histogram, we will have a more positive growth rate. In other words, the histogram is going to be more tilted to the right. And then if we retrieve skewness and kurtosis, those are very conventional histogram features, then skewness kurtosis in the progressive ADPKD are going to be positive. On the contrary, if the number of cysts growing are about the same with the number of cysts shrinking, and then the histogram is going to be about the same left and right, and then skewness and kurtosis are going to more convergence to zero in the stable disease. So we thought that perhaps we may be able to differentiate the disease severity based on skewness kurtosis if we can retrieve the reliably histogram of individual cyst growth rate. So 77 patients having mild moderate ADPKD, GFR measurements for 12 years, and then 3 years follow-up MRI scans were selected from the CRISP cohort. And we applied image processing technology, such as first, we segment the entire kidney volume using semi-automatic thresholding technique. And then we use the same method to segment cyst volume in the imaging. And then we co-register 2 images, baseline and 1 year after the baseline imaging. And then we labeled each individual cyst and then tracked the volume change of each individual cyst. And then histogram features automatically extracted out of it. So we use LabVIEW-based software. This is lab-made LabVIEW software, and then each step has a different front panel, but this is the last step. It shows this LabVIEW software shows the baseline images cyst labeled differently. So different color shows different number. And this is 1 year follow-up, and this is the fused one. You guys see my cursor? Okay. You guys see my cursor? So this is,
we can.
Okay. This is a fused one. So some of the cysts are increasing, some of the cysts are a little bit reduced, some of the cysts are about the same, some of them disappear, some of them newly formed. First, we evaluate our technology. So I obtained the information of HTTKV from CRISP investigators, and I compared with the HTKV value that we calculate using our software, and they have a very strong correlation to each other. Intra-class correlation coefficient, which shows data agreement, was 0.91. And then we measured measurement repeatability. So we used 3 patients for 4 times baseline, including the 3 different follow-up. And then we repeatedly measured. So in kidney volume, we used 12 kidney images and we repeated it 3 times and calculate the repeatability based on within coefficient variation. Kidney volume assessment about 10%, cyst volume 13%, cyst number repeatability was 6%, and squamous cortices were 12 and 24% respectively. So the repeatability using our software was about 10 to 20%. So this shows the volume rendering of the kidney volume and then cyst, and at the baseline and then each follow-up. Again, we were able to see some of them were disappearing, some of them grow faster or slower. And these 2 graphs shows when the patient have decreasing GFR change over time. And then when we have decreasing GFR change, the patient have more likely longer tail, and the histogram features like squamous cortices were larger than the other case. This is the case when the patient have relatively higher GFR change, and then an histogram was much shorter tail, and their squamous cortices much smaller. So let's look at the over 77 patient data. So this is the histogram, x-axis the histogram index, skewness for 1 year change, and then annual GFR change, and it was significantly correlated to each other, not only skewness, but also cortices as well. And also skewness was significantly correlated with the change of HTTKV over 3 years. However, cortices was not correlated, but barely not correlated, but skewness was. Let's look at HTTKV. So baseline HTTKV was significantly correlated with the annual GFR change. However, the total volume change over a year, over 3 years, were not correlated at all. So using this data, we can conclude that we propose the novel histogram-based imaging biomarker of ADPKD progression to complement HTTKV for monitoring the disease activity. So we saw that baseline HTTKV works fine as a prognostic imaging biomarker. However, the change of HTTKV may not be ever used as a reliable surrogate imaging biomarker. So for novel drug development, perhaps our imaging biomarker may play a significant for therapy response assessment. And also, I'd like to point out that the CRISP cohort imaging were not actually optimized for cyst segmentation. So sometimes I saw they use the pre-breeding imaging. Sometimes they use the breath of hold imaging. Sometimes there are contrast changes over the different institute, 4 different institutes. So I had to use different threshold to segment the kidney and then cyst. And I do believe that we can do much better job to standardize protocol. If we really believe that individual cyst tracking method is going to be useful. And then I believe that the biomarker accuracy will be even further improved than now. And another thing that I would like to finally, one more thing that I want to point out is that this approach is going to be most optimal for young patients. So in order to segment the cyst and then track it down, we need to have limited number of cysts. If the kidney has too many cysts, then it's really hard to segment individual cysts because they start lumping to each other. So I believe that these technologies would be most useful for young patients in having more limited number of cysts, and we may be able to more accurately track it down each cyst, how much is going to be changed over time. So I expect that prospective study with young patients under the clinical trial greatly helpful to evaluate the clinical utility of our strategy. So this grant was funded by a PKD Foundation research grant and NIH U54 and P30. Thank you for your attention.
Excellent. Thank you, Dr. Kim. So interesting. All right, we have one more. Our final researcher is Dr. Cynthia Sieben. Dr. Sieben is a postdoctoral research fellow in the laboratory of Dr. Peter Harris at the Mayo Clinic's Division of Nephrology and Hypertension Research in Rochester, Minnesota. A PhD graduate from the Mayo Clinic Graduate School of Biomedical Sciences, Dr. Sieben has had a long-standing interest in elucidating the mechanisms of human disease and in PKD specifically. After completing her doctorate, she's chosen to return to PKD research, and her primary research project aims to increase our understanding of the genetic complexity and pathomechanisms of ADPKD. Dr. Sieben?
Thank you, Elise. Okay, thank you very much. Thank you for the kind introduction, Elise, and thank you to the PKD Foundation for giving me the opportunity to present our research here today. And before I get started, I just want to say thank you to all the co-authors that contributed to this work. So the title of my talk is Comparative PKD1 and PKD2 Missense Variant Profiling, AIDS Molecular Diagnoses across the ADPKD spectrum, and reveals common pathomechanisms. Please note the PKD Foundation's activity disclaimer, and also note that none of the presenters in this session had a conflict to disclose. Elise gave a nice introduction for me, so I don't need to go into my bio further. And I just wanted to provide some background on some terms and definitions for my presentation. So an allele is a different form of the same gene, and these arise from variants. A phenotype is a set of observable characteristics. Pathogenicity is the quality or state of being pathogenic and the potential ability to produce disease. Pathomechanism is the mechanism by which a pathological condition occurs. And a non-truncating variant is a genetic variant that does not truncate or shorten the gene product or protein. And a missense variant is a genetic variant that contains a single base pair substitution in the DNA, which leads to a single amino acid substitution in the protein. So there are two main goals of my research project. The first is to determine the pathogenicity and pathomechanisms of PKD1 and PKD2 missense variants across different ADPKD subtypes. And the second is to evaluate the therapeutic benefits of enhanced PC1 or PC2 folding on ADPKD outcomes. So polycystin 1, PC1, or polycystin 2, PC2, both of these are encoded by the PKD1 and PKD2 genes. As you can see in the diagram here, both of these are rather large transmembrane proteins, with polycystin 1 being much larger than polycystin 2 and having a large extracellular domain, which is cleaved here, but actually remains tethered to the membrane fragment. The two proteins actually interact, and as you can see here from our previous work, we have found that polycystin 2 is required for polycystin 1 maturation and trafficking to the cell, to the apical plasma membrane, which will be a key feature that I'll discuss further in my presentation. And this you can see here in this IF image. And just as a note, so here I'm noting the missense variants, or several of the missense variants that are found in both genes and their locations within the protein. So as you can see, there are quite a few, and they are spread all throughout the protein. So currently we are using a bioinformatic approach, which is somewhat manual, to classify pathogenicity of PKD1 and PKD2 variants. And we use criteria as shown here. And with this method, we are classifying variants into 6 independent variant strength groups, or VSGs. And I guess the groups overall range from the most severe, being this group number 1, which are the fully penetrant truncating changes, down to the changes that we believe are likely neutral, which is the category 0. And as Alex mentioned in her presentation, the penetrance of these variants, or the severity of these variants, correlates with disease severity, as you can see in this graph here. So PKD1 truncating changes have the most severe, patients have the most severe disease, whereas those with PKD1 non-truncating changes have a later onset of end-stage renal disease. So although our variant classification by bioinformatic analyses is fairly efficient, we aim to develop a cell-based method to assess pathogenicity of PKD1 and PKD2 variants. And I'll just go over the workflow briefly here. So cells were isolated from the cyst lining of an AD-PKD patient. And then we transfect those cells with PKD1 and PKD2 plasmids, as shown here. We then label the cells with an mTERI antibody, which detects surface polycystin 1. And then we can analyze single cells, analyze the fluorescence of single cells using flow cytometer. And our readout here basically provides us with the information of the efficiency of polycystin 1 trafficking to the plasma membrane. And as you can see here, we can detect double positive cells, or those cells that are expressing both PKD1 and PKD2 with our fluorescent tags. And we can also detect the amount of polycystin 1 on the surface. So using this assay, we have assessed the pathogenicity of a number of variants across the different AD-PKD subtypes, and I'll talk about them individually. So for PKD1, we first assessed pathogenicity of monoallelic variants. So these are the typical dominant inheritance, where one variant is inherited from one parent. And as you can see here, overall, these missense variants typically lead to a fully penetrant phenotype associated with polycystin 1 trafficking. So here I'm showing surface polycystin 1. And we've included a couple truncating variants here, as well as some likely benign variants to test the efficiency of our assay. And I guess the take-home message here is that our bioinformatic analysis is fairly consistent with our cell-based method, actually. So we see the more severe BSG2 group contains only fully penetrant variants, whereas our milder VSG3 group has a range. And some of these milder variants, I provided some additional information below, including the location and the protein. There are several cases of complex inheritance in AD-PKD. So this is one example for PKD1. So in this case, we have multiple variants that are inherited from a single parent or from one of the parents. And this we call in cis monoallelic inheritance because these two variants are occurring in cis on the same chromosome. And as you can see here in these two examples, two of these variants, or in this case, 3 of these variants, cooperate to increase the penetrance of the allele. So you can see that these two variants initially are mild on their own, but in combination, they lead to a fully penetrant allele. And in these two families, we also have inheritance of 3 different variants in the same allele. And in this case, you can also see that these 3 variants on their own result in a milder phenotype, but in combination lead to a fully penetrant allele. There are also complex cases of inheritance, such as recessive or biallelic inheritance that occur in AD-PKD. So a biallelic inheritance case or recessive inheritance is when both parents have a variant in their in a particular gene, in this case PKD1. And both of these variants are inherited by the child. So I'm just giving a couple examples of these here. So in both of these cases, there is a missense variant that is inherited in PKD1 in combination with a truncating mutation, which leads basically to no function, which is similar in this case as well. And you can see that the phenotype of these patients varies quite widely from a very early onset to a early onset type disease. As a whole, you can see that these variants are much milder than the monoallelic and in-cis monoallelic variants, in that the majority of these, or all of these, actually have an intermediate effect, which is consistent with the hypomorphic or partially penetrant nature of these alleles. Interestingly, when we look at monoallelic PKD1 variants, we actually see that the majority of these variants are also fully penetrant when we use polycystin 1 trafficking to the surface of the cell as a readout. And here, I guess one important thing to note is that the majority of VSG2 variants that we have, so the more severe class of variants, are largely fully penetrant, but we do have some cases of milder variants as well. Whereas the VSG3 group, the milder variants, is more of a mix with several variants that are milder. And just as a note, which I don't think I mentioned before, these milder variants could potentially be impacting other disease pathomechanisms, and that's something that we're investigating further. So in order to determine the mechanism underlying defective polycystin 1 maturation and trafficking, we chose to evaluate protein folding. And protein folding is just, as depicted here, is just the process of the protein folding into its proper conformation. And the reason why we assess this is because missense variants can perturb protein folding and lead to degradation or conformational changes. And in order to assess this, we used a temperature shift with our culture system. And we did this because a reduced cell culture temperature slows cellular processes and increases the expression of chaperones, so improves the folding conditions of the cells. And here I'm just showing the workflow that we use. This is the same workflow that I showed previously. However, in this case, we're culturing cells at our normal temperature at 37 degrees Celsius and also at a lower temperature at 30 degrees Celsius. And you can see from a readout from this analysis, we see an increase in polycystin 1 on the surface of the cells, as well as in the percentage of cells that have surface polycystin 1. And this is for the wild type situation. When we look at our variants, so for PKD1, we see that a number of these variants, so here I'm showing the 37-degree condition, so the normal temperature in black, and the reduced temperature in blue, which is 30 degrees Celsius. And as you can see, there's a number of these variants that actually are folding variants and are impacting the folding of PC1. And at a reduced temperature, they have improved trafficking of polycystin 1 to the cell surface. And just as a note, the incompletely penetrant variants on the whole are temperature sensitive or are folding mutants. But we also see some of these fully penetrant alleles, both in the monoallelic and the in-cis monoallelic case, that are rescuing polycystin 1 trafficking, either partially or completely. When we look at the PKD2 variants, we see something similar, where a lot of these variants also improve, a lot of these variants also impact PC2 folding. And when we culture cells at a reduced temperature, we can actually rescue the PC1 trafficking to the cell surface. And there are several of these variants that are actually fully penetrant and are rescued to the wild type levels at 37 degrees Celsius. So in summary, the majority of the monoallelic variants, both the in-cis and the typical monoallelic for PKD1 and PKD2, these variants impact PC1 trafficking to the cell surface by greater than 80% and are fully penetrant, the majority of them. And the biallelic variants have intermediate effects and are much milder. Overall, our bioinformatic predictions in our in vitro or cell-based analysis are quite consistent. And many of the PKD1 and PKD2 variants that we've assessed disrupt PC1 surface localization due to defective protein folding. And this can be partially or fully rescued at reduced temperature. And then just as a final note, defective PC1 surface localization, we find represents a common pathomechanism in ADPKD. So the main impacts of our study, we believe, are that the development of an improved method for determining variant pathogenicity will be of diagnostic and prognostic value for patients, clinicians, and researchers, and that identification of common pathomechanisms creates entry points for therapeutic development. And also, we believe that targeting proper PC1 and PC2 folding for ADPKD therapeutics may represent an effective treatment, but this still requires further testing. So I include here my contact information as well as that of my mentor, Dr. Harris. And I think now we will transition into the Q&A.
Excellent. Thank you very much. That was fantastic. All right, I have a couple questions. Let's start with Alex. So you did some really great work with data sets that we saw that highlighted a lot of disparities. And I know that you also do some work interviewing patients directly. So could you share with us what it's like as a researcher to have two different kinds of data from a medical record or data from directly from a patient, and how that kind of changes the conclusions that you make?
Yeah, so I'm also a research assistant for the Hospice Project at UChicago Medicine in the section of hospital medicine. And it's a different research project, but it definitely gives me some perspective on what the motivation behind my research is in racial and ethnic disparities in PKD specifically. And I think I did both these research projects this past year, and it was just really rewarding to be able to do the research to hopefully eliminate these disparities down the line by contributing to further research on them, as well as actually interacting with patients, not necessarily PKD patients. These were just general section of medicine patients. But it was really rewarding to be able to hear from their perspectives personally on their own care, their own experience with the Chicago medicine hospitals. And it really did ground what the aim of my work is to reduce these disparities and improve patient care at the end of the day. And that only comes with really understanding patient experiences by actually talking to them and actually listening to them and being able to be a resource and advocate for them. So I think they complement each other really well. I'm not sure if that answers your question specifically.
No, that's perfect. Yeah, it was just interesting to see that, one of your data sets was over 40,000, I believe. And then you have smaller data sets of 3 or 4, and it must be interesting to see the differences.
Yeah.
Great. And there's also a question about the definition of a racial or ethnic category. How do you handle it if someone has 2 or more?
So I used the USRDS data set, and basically USRDS is the United States Renal Data System. And so they're a national data system that collects, analyzes, and distributes information about CKD and ESRD or kidney failure. And so I did a longitudinal study retrospectively from 2000 to 2018. So some of the variables they have one for race and ethnicity. Ethnicity is a yes, no, Hispanic or Latino, or not Hispanic or Latino. And then race has white, Native American, Pacific Islander, Asian, Black, or African American, as well as others, including multiracial. So the reason why we focused in on non-white African American patients, non-Hispanic African American patients, non-Hispanic white patients, and Hispanic white patients is because there's such little literature on ADPKD racial and ethnic disparities, and we wanted to focus in on the groups that we felt were going to be most burdened based on other research and other kidney diseases. So those that most came up were the disparities across white and African American patients, as well as white and Hispanic patients. So we did look at the other racial categories. We initially had African American patients, white patients, and Hispanic patients, and we grouped in the other categories with the white patients, but it didn't feel right to group all in those races together because we didn't want to lose their stories and narratives. So we just focused in on the groups that had the biggest disparities across them. And when we looked at Asian Americans, for example, I saw a comment on that the disparities weren't that great. And then some of our models didn't actually have disparities. But yeah, in further work, when we're working on publishing the manuscript and things like that, we're definitely going to consider including those as well, just to show and make sure that no one's voices aren't heard in our analysis.
Fantastic. Thank you. Yeah, that was great. Dr. Kim, so
yes.
Talking about tracking individual cysts is really interesting. And I like what you said about how you need to probably focus first on younger patients who have more cysts that are kind of by themselves so you can easily track them. Could you talk about what that study would look like and what participation would look like for patients?
I think that probably if there is a study of novel drug development, and then we may be able to recruit a patient within certain age or certain category, like a mild disease. There are not that many though. So young patients, like a CRISPR cohort, the young patients were not that many, but we will be able to identify young patients so that we will be able to apply our strategy and then see how effective it will be. It's going to be, it should be large study though, because the patients, what we are focusing on in CRISP cohort and the Holt cohort, they are all like advanced disease. So it was a little bit hard to think about whether we have to apply this strategy and how it's going to appear in young patients. But if we will have to design prospective study focusing on more young patients, then we will be able to have a very interesting conclusion whether this strategy is going to be effective or not.
These would have to be patients who are either on a therapy or in a clinical trial?
I think so, because I personally think this biomarker is going to be most effective for therapy monitoring, because we are looking at a change over a certain period, and the current total kidney volume based indices were not actually too much effective to measure the change of the kidney, because it takes really long time. Baseline HTTKB means actually the change of TKB for like 10 years or so, 10 years, 20 years, basically that's what it means. So like a 3 or 4 years tracking may not be able to that accurately predict the renal function. So in order to evaluate new drug, we need to develop new surrogate imaging biomarker. And in order to do that, I think this new approach is going to be useful. That's what I'm believing in.
Yeah, it's great to have a new tool to really understand how therapies are affecting different kinds of patients. So we're glad you're working on that. Dr. Steven, last question for you before we wrap up. You did a really great job in your last slide about talking about what this impact of patients might look like. Could you share with us kind of what happens? So once we understand how these variants and mutations affect the disease, what's next when we start testing compounds or therapies? Is that, what does that look like, and how long do you think it'll take to get started?
That's a great question. I wish I had a clear answer for that, but so basically once we've identified a common pathomechanism, which we have here for PC1 trafficking, now we would try to find compounds that improve protein folding conditions in the cell overall, or potentially doing a compound screen to look for targeted compounds that improve PC1 trafficking. So our next steps for this project are to test various compounds. So there are a number of chemical chaperones that are available. So we would test those compounds in our cell culture system, and then we would move on to an animal model system. So we have a mouse model that has one of these missense mutations in PKD1. So we would do that testing. And then from there, from the animal part to a clinical trial. I'm not exactly sure what the steps are, but I think that's a longer process.
Yeah, no, that's great. Thank you. It's helpful to understand how you go from looking at, those mutations to moving into animal studies. And the next session actually is about clinical trials. And so we'll be talking about what that pipeline looks like and what it looks like to get something into a human study. So stay tuned. Thank you to all of our speakers so much for joining us. This was fantastic. We really love to see what you're learning and the research you're doing. And thank you from the foundation for doing such great work.